All right, looks like everyone's in, so I'll get started. Hello, everyone. Thank you again for coming to one of our Funk Biogeography seminar series. We'll get started in a second, but I have a couple of announcements first based on things you can do to get involved in TUBES if you're possibly interested. First one is that we are currently looking for a few people to join our TUBES education committee. So that's been active in a few years. We're figuring out the role of that. If you'd like to help get involved with getting that set back up and deciding what this is going to look like in the future, we'd love to have your help. Um, we'll have you contact Michelle Lawing, alawing at tamu.edu if you're interested. And all the things I'm going to talk about will be available on all of our social medias, Blue Sky, Facebook, also our website at biogeography.org. So if you want to see this again in the future, just go into one of those and you'll be able to find all this. The other one, slightly more convoluted. We're currently offering funding for workshops. So we have between 500 to 1,000 US dollars in financial support for organizers of workshops broadly in biogeography. So the criteria are fairly straightforward. Time period would be workshops between October 2026 and November 2027. This can be in person or virtual. We tend to encourage virtual because it lets the most people participate, but either is doable or hybrid. And as far as content goes, broadly we're looking for methods and approaches in biogeography, but any related topic is welcome. So some examples of things we've done in the past include species distribution modeling, bioinformatics, Bayesian statistics, evolution through time and space, biogeography of stress, and plenty of other ones. So if this sounds like something you might be interested in, the submissions for that are fairly simple. What we have will be some contact information for the organizers. If there's going to be multiple organizers, just let us know who to contact specifically, and some details of the workshop. So when do you think it's going to take place, and what are some details about the content and who it's intended for, any requirements you might have as far as room or power. And if you have one of those to submit, we'll have you submit that to Mark Costello, conveniently today's speaker, at mark.j.costello@north.no. If you could do that by the end of next month, so by 30th of August, that would be great. And like I said, that's also available on our social media, so if you need to come back to that information in the future, it'll be available. So as far as the seminar itself, the structure of this, it's going to be about 45 minutes for our speaker to talk, and then 10 to 15 minutes of Q&A. So if at any time you have a question you want to submit, the easiest way to do that will be to go to the Q&A button at the bottom of your screen. You can type in a question there at any point, and at the Q&A session at the end, we'll go ahead and read that out for you. And as always, looking ahead to August's speaker, our next speaker is going to be Carlos Daniels Cadena Ordonez from the University of the Andes. So those details will be out sometime in early August. Keep an eye out for that as well. And always, thank you so much for coming to one of these. If you'd like to see it again in the future, or if you weren't able to arrive day of, these will be uploaded on our YouTube channel at International Biogeography. If you want to become a member of the society, make a donation, or just kind of keep an eye on what we're doing, I'll direct you to our website at biogeography.org, and also our journal, Frontiers of Biogeography, at biogeography.pensuff.net. It's a nice constant stream of interesting biogeography content out that way. And after that, I will hand it off to Marta to introduce our speaker. Thank you.
Thank you, Hikaru. Welcome back, everyone. I am Marta Jarzyna. I'm the Director for Public Affairs and Communications at the International Biogeography Society. I'm excited to be back after our June break. Just FYI, our speaker for June, Julie Aleman, we will have her back in 2027. So we didn't cancel without a plan in the future. So stay tuned for that. But today it is my great pleasure to introduce Dr. Mark Costello. Dr. Costello is a professor at the Faculty of Biosciences and Aquaculture at Nord University in Norway. He received his bachelor's degree in zoology from the University of Galway and his PhD from University College Cork, both in Ireland. And following his PhD, he completed a postdoctoral fellowship at the Marine Biological Association in Plymouth, held jointly with the Marine Laboratory in Aberdeen and Napier University in Edinburgh. He then spent 6 years as a lecturer at Trinity College Dublin before becoming a professor at the University of Auckland in New Zealand, which he was for 16 years until joining his current institution in Norway, Nord University, in 2020. Mark's research interests span biogeography, biodiversity, ecology, particularly in marine environments, with applications to conservation, climate change, invasive species, and all kinds of applied topics. And today we'll hear about parts of his research and specifically about the latitudinal diversity gradient, I presume, in marine environments. So please help me welcome Dr. Costello. Mark, the floor is yours.
Thank you very much, Martha. Yeah, so I'm feeling a bit embarrassed after that long introduction. But actually, Galway is where I first learned about biogeography in 1982, before most of you were born. And a lecturer named T.K. McCarthy explained the theory of Ireland biogeography, and I was amazed that there actually you could put general patterns to nature because when we look at nature from our own point of view, everything seems so chaotic and unpredictable, and we're always looking at more local ecology effects. Um, so that meant I went on and did my PhD in biogeography, working in marine systems. I shifted from freshwater to marine. And that kind of led to my interest. So once I got an academic position in New Zealand, I had the freedom to pursue my own interests in biogeography, and this led to the present talk. And it also, so I'm going to talk about three things, including some unpublished terrestrial latitudinal gradient work. Um, and I'll ask, why does species richness peak at the equator, or does it? Maybe it doesn't, and what drives this gradient. And then a paper we published in the Frontiers of Biogeography, the best journal in the world because it's our society journal, um, on why 20 degrees is significant, at least for marine species. And we all know that probably you've come across this latitudinal gradient. It's in all the textbooks, but it's presented in different ways. And some of these ways have been misleading and misled many of us, including myself, until recently. And one of my former PhD students, Hani Saidi, was interested in razor clams, which don't actually occur in New Zealand. So she had to look at data from different sources. So she got latitudinal data, went visited museums around the world, improved the taxonomy of the specimens, and we found this surprising dip. So on the axis here, it's going from the north to the south. Some of the graphs go the other way later. Um, and you can see the number of species is a solid line and this big decrease around the equator. So we thought this was very surprising and due to taxonomic effort or sampling effort. But actually, when we checked the species, when she checked the species in the museum records, the dip became more pronounced. And it seems that, at least for this group of species, they don't really like it too hot around the equator. So you end up with these peaks. And the dotted lines are the average sea surface temperature, which you can see is as you predict, is correlated with latitude. Um, so that was quite interesting. So she went on and gave a talk at one of the Biogeography Society conferences. It was well received. The editor of the journal asked her to submit her paper, and it was accepted, but with one exception. So they said that she shouldn't mention that the gradient is bimodal in the title and not mention it in the conclusions of the discussion. So we were a little bit surprised because we thought this is the most remarkable finding. And just for razor clams, it may be just the way they are, because we know that some groups of species, like penguins, for example, only occur in the southern hemisphere, and other animals only, species and plants only in certain parts of the globe. So we didn't think it was that surprising that one taxon should have an abnormal latitudinal gradient. Um, but I'd had a copy of the Ocean Biodiversity Information System dataset. So I quickly looked at that, and the next graph is what I found. So this is for all kinds of species collected in all kinds of different ways. So you would expect it to be fairly chaotic data. Um, and so each of these dots here is a latitude, longitude cell. The number of species on the left, just the number counted. So you can see incredible variation, at least in all latitudes, there's some species. And then the white arrows show that we have high numbers of species in the northern hemisphere, which is probably due to sampling effort, but also some in the southern hemisphere. Um, But we also see this decrease then in the higher latitudes as people expect. But what was very surprising is this dip here is around the equator as well. So why would there be less sampling, particularly at this point? So we puzzled about this a little bit, but it's clearly bimodal. But this is using what you'd call alpha diversity, the number of species in individual samples. What Hani was doing in her samples was looking at gamma diversity, which is the total number of species in the latitudinal band. So as we didn't really want to change the title and the conclusions of our paper, we then had a look at more data, and we went through the literature. There were about 60 papers looking at marine latitudinal gradients, and every single one of them had a dip at the equator. We couldn't believe it. And only one of them commented that the dip might be real. The others just drew a line straight across and assumed that this was a lack of sampling at the equator or something. So we thought, well, this is kind of surprising. You couldn't find the same pattern across so many different studies in a big global database and for these groups. So on the graphs here, we have the average number of species, which is alpha diversity, the total number of species, the gamma diversity. This is for all marine species, a sample about 75,000, which at the time was about there's around 250,000 marine species. So it's a pretty good sample. And then if we take the estimated number of samples, species per 50 samples repeatedly, you can also get this rarefaction method called ES50. And in all these cases, you always get this dip at the equator. And this is exactly what people have predicted from climate change. So we thought, well, okay, it's not so surprising, and there could be a mechanism behind this. So that we sent them the draft of this paper. In fact, we published this in Trends in Ecology and then cited this to the editors of the other journal, and they accepted the paper title. So I've mentioned alpha diversity and gamma diversity. So the other kind of diversity is beta diversity. And Chaya, who did that previous paper in Trends in Ecology and Evolution, she looked at beta diversity afterwards. And you can see that the solid line here is beta diversity, and it kind of peaks around the equator, whereas the gamma diversity dips. So it has this opposite relationship. And we attribute this to the high species turnover around the equator because of probably climate change is causing many species to move away from the equator or to decline there. So you get this high turnover. But that's just our hypothesis for that. And as far as I know, nobody else has looked at beta diversity gradients with latitude in marine or other systems across the whole globe. And we also know that climate change is changing species. So I'm living now up in the Arctic, Norway, at the top here, just inside the Arctic Circle, which is the dotted line. And one of our studies by Cesc Gordo-Villaseca, his PhD, he looked at species changing in this area here. So he had nearly almost 200 fish species from standardized fishery research trawl data over 30 to 40 years. And he found that there was this huge increase in species up here in the Barents Sea in this part of the Arctic Ocean. And what's interesting is the graph on the left, where species richness is the black line and sea temperature is the red line. And you see the temperature scale goes from kind of 1 to 2 degrees here. But almost in synchrony with the sea temperature, you get this change in numbers of species. So what we're really sampling is species activity. The species must be there all the time. They're not all kind of suddenly moving and back and forth every year. So this is quite interesting that there's an immediate response of marine species, apparent abundance and presence due to climate change. And this might make the ocean a little bit different than terrestrial systems where species movements like this may not be so immediate and in such a good response to warming. So he actually found there's 2 times more species in the Arctic. Somebody actually said you shouldn't say that. It sounds like good news. So I don't know, but that's the way it is. And was highly correlated with sea bottom temperature. And just in case you're interested, it's not causing Arctic species to disappear at all. In fact, some Arctic species are increasing in their abundance. We have abundance data as well. And some are decreasing. But it looks like the Arctic is already too cold for some of the species that live there. So they like an extra degree of warmth is pretty good for them. But looking more at climate change effects over long time scales, there's this amazing data set on sediment cores all around the world. So on the maps here, each of these dots represents a sediment core, and within that core, they can count, for example, in this case, the shells of planktonic foraminifera that settle down from the plankton and rest on the seabed around the world. So you get this abundance on a very crude time scale of the orders of thousands of years, or at the best, hundreds. And when we look at the depth of the cores, if we look at the last glacial maximum, which is the top graph, and this is the latitudinal gradient, you see maybe a slight dip around the equator. And you look at pre-industrial, which is kind of recent centuries, before the 20th century, you see this dip increases. And then this is predicted to decrease further by the end of this century. So we can see here that the latitudinal gradient, at least for planktonic foraminifera, is also bimodal now, and it's predicted to become more bimodal with climate warming. So what's the pattern with present biodiversity data? Well, Chaya went on to finish her PhD, and this was the paper she produced from that. And here she used the Ocean Biodiversity Information System database again, just for animals in this case, because the plant data was not consistent enough. And we know the animals better. So if we look at the gradient before and after 1985, in the published paper, actually, we showed 3 time periods to show this better in a way. But just to keep it simple here, around early 1980s, all sea temperatures were warming almost everywhere in the world. Whereas before that, the change in sea temperature was much more variable spatially. So we chose that year, and the information's in the supplementary material as to why we did that. So the red is before and the blue is after 1985. So we can see that these peaks in the subtropics are increasing after 1985, and that there's a decrease around the equator. That's for all species. And if we look for seabed living species, we see the same general trend. And if we look at pelagic species, the trend is even more pronounced. So pelagic are the open water species that can move immediately in response to the water masses that they're living in. So maybe they don't know they're actually moving. They just stay in the right temperature for themselves. And so now we have evidence that the latitudinal gradient in marine species has been decreasing since the last glaciation, about 20,000 years ago, when it was almost flat. And this decrease at the equator and probably increases in the subtropics are enhanced now with recent global warming. And I've got a few arrows here. We also see that the shift in the northern hemisphere, which is on the right of the graph, is more pronounced. We don't see such a shift in the southern hemisphere. And the same for benthic, and the same for pelagic species. But there may be other factors going on here as well. So it's not simply, even though it's kind of amazing that you get whole groups of animals like penguins in the Southern Ocean, but we still see these general trends in species numbers that are the same. And I think it's amazing that food webs seem to end up being constructed in a similar way with similar numbers of species, even though you might have completely different evolutionary groups of species in different parts of the world. I mean, this has been known for a long time, but I still think it's amazing. So when we look at photographs of the tropics, and I remember David Attenborough pointed this out, when he goes onto a coral reef, you see fish and life everywhere in the water. When you walk into a tropical rainforest, you see hardly anything moving. It's just the trees and the plants, and all the animals are hiding. But so in the tropics, this is Raja Ampat in Indonesia, which is the highest endemicity of anywhere in the oceans in the world. You see the fish are just a huge abundance in front of your eyes. But when you go to the colder areas, such as Channel Isles of California, up here in Norway, or down in Chile, you don't see so many fish. But what you do see is huge numbers of benthic invertebrates walking or sitting on the seabed. And you don't see that many visible benthic invertebrates in the tropics. And I suggest that this is because fish predation in the tropics is so intense. that it's very hard for benthic invertebrates to show themselves. They have to stay hidden away underneath the corals or in the sand. Um, and fish predation and fish herbivory only really occurs in a big way in the tropics as well, at less than 20 degrees centigrade. So fish dominate the tropics to such an extent that they may have pushed out invertebrate species. And the Reef Life Survey is a standardized global survey using scuba divers on coral and rocky reefs around the world. It's over 2,000 sites. And we published a paper a few years ago looking at this data. So here latitude goes from 0 to 80 degrees, both the northern and southern hemispheres are plotted together. But what we see is that for species richness of vertebrates, which is mostly fish, but also includes the odd seal and turtle, it decreases with latitude. But large mobile invertebrates like crabs, lobsters, squat lobsters, starfish, sea urchins increase with latitude, and even their abundance also increases. So here we have an inverse latitudinal gradient for some of these invertebrates, maybe in response to not being able to compete and survive against the predation of fish species. But what about terrestrial latitudinal gradients? So I kept following the terrestrial literature on this and emailing people who seem to have data sets. And I asked them, had they looked at the latitudinal gradient in their species groups? And probably emailed 5 or 6 people. And they all said, no, there's no point in doing that because we know the pattern, which is exactly what the marine people were saying 10 years ago, where all these papers on marine latitudinal gradients assumed the gradient peaked at the equator, so they just drew a line straight over the top. So I thought, well, okay, maybe with my colleague Chenshur Zhao in China, in Qingdao, I said, well, maybe we could look, would he be able to run the data through the Global Biodiversity Information Facility and see what the pattern is, just out of curiosity. So we were able to get 0.6 million terrestrial animal and plant species. So this is the table of the species here. It's important to notice that insects and plants make up nearly about 90% of all the species in this data set. So you'd expect to get more general patterns from the groups with more species, and the groups with less species, like the earthworms and reptiles and amphibians, you may see more variable patterns because you have less data. And this is all kinds of different data sets collected in all kinds of different ways. So if you find any general patterns, it's kind of, I think it's amazing. And there's other patterns that we know that are very important in the world as well. There's two that we included in the paper that we had submitted. One is human population density, which would have a positive effect in maybe increasing sampling effort around where people live, and may have a negative thing in actually pushing some species away, but probably not causing whole regional extirpations of many species. And also remember that most species are insects and plants. So a lot of the time we have mammals and other charismatic birds species in our minds, but they don't make up so many of the species. And another interesting classification of ecological land units, which is available in the ArcGIS platform, classified land cover around the world into different climatic units. So these kind of bring together all that environmental information on climate into one kind of metric, because otherwise you're dealing with so many different hypothetical environmental variables which are intercorrelated. You could do lots of analysis and just get even more confused. And if we look at these, we divided also, I meant to mention, we also divided the world into 3 different longitudinal zones: the Americas, Europe, Arabia and Africa, and the Asia Pacific, just so we have kind of 3 replicate samples, and also because the computational demands make it easier to subset the data. So if we see the same general pattern across all longitudinal regions, it gives us some confidence that there's something fundamental like temperature or day length which is driving the gradient. And we see that temperature is the same gradient across more or less all 3 longitudinal regions. The ecological land units are also similar, but interesting that they actually dip near the equator, which is the green ones here, and then the green ones went gray on this graph. So there's little dips, but and there's another slight decrease here. So there's slightly less ecological land units around the equator. This might be because most of the big mountain regions are not on the equator itself. They're north and south of the equator. And there were several talks at the Biogeography conference in Aarhus, which pointed out this high diversity of mountainous regions in the Andes and in Asia and in Africa, which were away from the equator. And of course, these mountainous regions then within a narrow uh, space include many different climate zones down the sides of the mountains. So they can actually include a lot more species per kind of unit area. Right. And then there's other patterns that and the land area also dips near the equator in the Americas and Asia, but it peaks in Europe and Africa. Okay. So we then just ran the data set and looked at how did the total number of species shift over time. And again, we looked at before and after 1985. And in both the south, which is the left bar, and the north, which is the right bar in each of these plots, we see that it always increased for all the different groups, for the amphibians, birds, clitellata, which is the earthworms, insects, mammals, plants, reptiles, and all the groups. In fact, it increases most for the earthworms in the northern hemisphere, which I find curious. I don't know why earthworms would move more than any other group. So if anybody has an answer, I'm pleased to hear it. And we can see that species have moved around, on average, the peak richness has moved around 167 kilometers before and after 1985. So we also expect this. Yeah, it's interesting that we just got the reviews back from our paper yesterday, and one referee says all this needs to be verified. You need, better standardized sampling, da 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 da. And the other referee, and so they're highly skeptical of all the results. And the other referee says, well, this is all obvious. There's nothing really new here. We knew that these patterns would exist. So you just can't win with referees sometimes. So if we look at all the latitudinal gradients, so then the three columns here, the Americas, Europe, Arabia, Africa, and Asia Pacific. The dashed red line is the equator, and we have the amphibians, the insects down here, the plants, and all species. So if we look at all species, we see that there's it's always bimodal. For the Americas, it's a little less so. But in different groups, you are getting this bimodal pattern. So maybe there is something here, and certainly I don't think you could say with only a couple of graphs that it actually peaks at the equator. Yeah. Okay. Next slide. So if we go back a bit and look at that planktonic foraminifera data sets, if we plot the number of species from the last glacial maximum, these are specimens and cores, and then from the recent centuries. So the graph, each of these dots is independent. It's very curious that we see that at 20 degrees centigrade, you get this decrease in species richness. And we thought that's pretty cool that you get it both at the last glacial maximum and in recent centuries. So probably for planktonic foraminifera, for some reason, they don't like it above many of the species don't like it above 20 degrees, and the warmer it gets, you get less and less species going on. But that could be just planktonic foraminifera. It may not be a general pattern. So went back to Chaya's data in the OBIS data set of about 50,000 species. And this is the overall graph on the right. And we see that species richness globally start reaching an asymptote at around 20 degrees. And for pelagic fish down the bottom left, it decreases after that. For benthic fish, it's much more variable. The variability increases between latitude, longitude cells, which are what each of these dots are. And benthic invertebrates, it also decreases. They're the seabed living invertebrates. And we could go into more details and look at other different groups of bivalves and gastropod mollusks, which are mostly benthic. polychaete worms, which are also mostly seabed living. The red and the black circles are the southern and northern hemispheres. So if we see the same pattern in both hemispheres, this suggests it's due to temperature and not due to other factors like area and so on. And we can see that with the exception of demersal, which are kind of near seabed and reef-associated fishes, they seem to like it a bit warmer, and their peak is around 26 degrees. So that's kind of interesting. But is that the same for terrestrial species? And definitely not. So this surprised me. May not surprise everybody, but here species richness peaks in all three of the longitudinal regions between 10 and 15 degrees. So it seems to be much cooler. But of course, we're measuring here is a species richness in the surface, the annual surface, mean surface temperature in a square cell or hexagon on the planet's surface. And some of those hexagons will include mountains, which would have a huge range of temperatures from freezing down to, in the tropics, maybe 30 or 40 degrees. So this is taking that average. So I don't really know why this is the case. So if anybody has ideas, this is another good answer, but it's definitely not showing the same decrease in richness above 20 degrees. So if we go back to the marine data again and we look at the Reef Life Survey, the standardized global set counted by scuba divers, and if we take the median temperatures for the thermal range of each species and then you plot them together, you end up with at least two different climatic gills of species, the temperate, which are the black, and the tropical, which are the brown cells here. But of course, these species overlap together. So where they overlap is the line here. So for fish on the left here, you see the peak in richness is again in the around 26 degrees, like it was for the OBIS data, which are completely a different data set. But for invertebrates, it's actually around 22 degrees, I think. So again, we're seeing this effect around the low 20s of effect on richness, even in standardized data sets. And then we started looking around in the literature, and we're pretty struck by the fact that gross phytoplankton production decreases above 20 degrees because the rate of respiration increases faster than the rate of photosynthesis. If we look at the oxygen vulnerability of species on the top right here, we see that above 20 degrees, you suddenly start getting a lot more oxygen toxicity. And this is oxygen on the left in milligrams per liter. So yeah, so 20 degrees. So the effect of low oxygen on species is not a linear effect. It's a curved effect, which starts really taking off after 20 degrees. So oxygen consumption is increasing then, and probably oxygen availability is less. And then a paper that came out just at the time as we were writing ours looked at standardized, they called them squid pops. These are little bits of squid meat that they stuck on a stick in the sediment, and they put a camera beside it, and then they measured how long it took for that squid to be eaten by fish in the area. And so this standardized method of looking at predation intensity found this peaked at 20 degrees for these demersal fish. And another study, which was published around the same time on pelagic fish, showed that pelagic fish predation on bait peaks at 20 degrees as well in the Pacific Ocean. So that's kind of cool. And then if you look at net seaweed, so up the top left, we have phytoplankton, gross phytoplankton production. Seaweeds also, at least for these species, their growth rate or net growth rate starts decreasing above 20 degrees centigrade as well. So we see all these strange patterns, but they could be just coincidences. So we see this is kind of a summary of the patterns, but then I'll get into a little bit about my thermal energetics. So we see that clearly species richness has been declining around the equator since the last glacial maximum. This suggests, and you look in the fossil record in our paper, we describe fossil data, which also shows species extinctions, marine ones, decrease, extinction rates increase above 20 degrees. So it looks like for all of life, at least in the ocean, temperatures above 20 degrees get less and less suitable. So of course we have species that can live into much higher and species that can live at much colder temperatures, but most species like it around 20 degrees, it seems. And this means that the equator is already too hot for some species, and with climate warming, those species will either have to move or they will be extirpated there. And we know there's many studies on individual groups of species showing species range shifts. Citizens can see this now with their citizen surveys of how butterflies and birds and other species and plants are changing their habits due to climate change. We also see that tropical and temperate species, their gills separate around 20 degrees centigrade, and that's where they overlap. You then get most species because you get both the temperate and the tropical species overlapping. And other factors like predation rates and photosynthesis. So we had a workshop with some colleagues from Tasmania, one of them who was a biostatistician and worked in thermal energetics. And it took us at least one workshop to understand all the jargon and the technicalities of thermal energetics, because the rest of us were just ordinary ecologists and biogeographers. But there's this general curve of everything that happens in life. I haven't tried to apply it to people yet, but it might apply. But if you look at temperature, and here it's just going from 0, -10 to 40 degrees, you always get this asymmetrical curve with things happening slowly at cold temperatures. Then they really heat up, and then if it gets too hot, things stop altogether. And these, on the left, I have a list of all the different things that could be a metric on the left axis. And in the literature, people usually refer to the peak of this as the optimum, the thermal optimum. But that's only the thermal optimum in a laboratory experiment where everything else is just lovely and the species can keep growing at that temperature. In the real world, if you're living that close to the edge, and you're at a very maximum temperature, so some functions may be maximized, but also you're taking a risk because you may run short of food and die, you may get eaten and die. So if you're foraging, you're exposing yourself to predation. And it just takes 1 or 2 hot days and you die. So probably many species around the equator are already living close to or above their thermal optimum. So any extra stressor is going to cause their death. Particularly, for example, in shallow waters, oxygen availability will just decrease when the warmer it gets. And so if you need more oxygen for a few minutes, that's just too bad and you die. So the real optimum is probably a bit less. So if we get into a little bit more detail. So as you know, everything is about loss and gain in our bodies. We have to replace proteins all the time. So people look at enzyme stability. So what is the most stable temperature for enzymes to function? So the optimum enzyme stability will then probably be a bit less than this thermal optimum, and certainly the fitness of the species would be a bit less than that. And okay, so this graph here is kind of leading the discussion a bit because it's showing that it's probably around 20 degrees. But of course, every species will have its own individual curve. And some species, their optimum will be less than 20 and some will be more. We're trying to look for a general pattern across all species. And about 10 years before we did this work, there was a review paper published in Proceedings of the National Academy of Sciences, and they reviewed about 1,000 species, and they found that around 19 to 21 degrees were the optimal temperatures for the marine and freshwater species from the literature. So we did that review again. We got around 7,600 samples, and this graph shows the model estimates from Ross Corkery's model, my colleague here. So the model estimates that around 20 degrees should be the most stable temperature. Actually, I'm missing, that's it there. And then if we look at the range limits for every species, because you don't really know, if you take the average, it's not necessarily the optimum temperature for a species. So it's safer to take the kind of thermal range limits, its max minimum. When you put all those together for terrestrial, marine, and freshwater plants and animals, you can see they all overlap around 20 degrees centigrade. And actually, the energetic models across all domains of life, the bacteria, archaea, and eukaryota, they all show optimum stability of temperature of energetics in cells at around 20 degrees. And there's actually literature on the biochemistry of cells showing that the optimum temperature for enzymes on average is around 20 degrees. So there seems to be some fundamental physical and even molecular basis for to why energetics should be kind of scaling from the molecular level right up to the cells, to the species, and in the slides I showed earlier, right up to the latitudinal gradient and these general patterns in biogeography. So we think we were pretty excited about this. So it seems that 20 degrees seems to be kind of this Goldilocks zone. It's not too hot and not too cold for life, that most species can live at 20 degrees, even though some evolved to live more and less temperatures. So I don't think there's been many other tests of this from other people. So I welcome other people to look at their own data sets and see if they find any similar patterns. And certainly the terrestrial latitudinal gradient with a much lower temperature needs further research to explain if because in our data sets on the literature, it showed that most terrestrial species like it at 20 degrees. Maybe the way we're sampling temperature in the terrestrial environment and reporting it in that draft paper are not quite right. And so that's my last question really is how this latitudinal gradient in terrestrial biodiversity is being driven. It's not is it temperature, but we're not measuring the temperature the right way. Thank you very much, and I'm happy to take any questions. Thank you.
Thank you very much, Mark. That was really interesting. There's a couple of questions in the Q&A box, which I will read now for everyone's benefit. Both of them are from the same person, so I'm just going to lump them together, I think. It's from Iram Abbas, who is disclosing that they're a first-year student in ecology, but I don't think that matters. So the question is, why can't land animals just handle 20 degrees Celsius like marine life does? What's the main reason that limits them? And the follow-up, between land and sea, which one do you think is closer to hitting its limits first under current global warming rise?
Yeah. The first question is about why can't land animals
Handle 20 degrees Celsius
yeah.
like marine life.
Actually, there's papers showing that the optimal temperature for people is 20 degrees. So office temperatures generally are around 20 degrees. They should be 18 really. We should just put a sweater on and save energy. But apparently for people working outside, it takes more energy for us to lose heat as it gets warmer. So even though we can live at much warmer temperatures, we need to be able to cool down. So this becomes stressful. And as people are explaining in the news in Europe a lot at the moment, that this stresses your body organs and eventually it exposes you to other diseases. So 20 degrees is also the optimum for humans and maybe for all species that are at the top of the food chain. Maybe the 20 degrees, they don't have to adjust their temperature to be warmer and colder because they're just in charge. Maybe we can do that. But in the terrestrial environment, it's much easier to cool down because you can evaporate sweat and you can go into caves and you can go into shade. You can choose where you live if you're mobile. So maybe in the ocean, it's a little bit more difficult for species to avoid a temperature change. The only exceptions are a few papers showing that under seaweed and seagrass and in caves and shallow waters, that the effects of heat waves are a bit less in the ocean. So that species that can just move down a little bit deeper or hide in the shade do save a few degrees. But that's like in the Mediterranean and really hot waters. So it is interesting as to why, and also many terrestrial species become nocturnal and avoid the heat of the day. Yeah. So this does get down to the ecology and the energetics of different species.
So between land and sea, which one do you think is closer to hitting its limits under current global change?
I think because marine species can move freely, they're doing okay
Hmm.
generally.
Yeah, there's
Certain
certainly, yeah.
Yeah, maybe some places like in the Mediterranean or some of the Asian coastal seas where species can't move out, then they will be extirpated within those areas. But this is again a hypothesis. Um, when we were writing the IPCC report, we so many papers said species are going extinct due to climate change, and we struggled to find any evidence. And then we realized most people confuse extinction and extirpation. Of
Hmm.
course, people in biogeography know the difference, but so they were talking about local extinctions, and the species may have moved. And they also never count the new species that arrive. They're always counting what we've lost, but not what we're gaining. Um, so I think these are interesting ways to look at it. Yeah.
Yeah. Okay. Next question from Marcia Barbosa. Great talk, thanks. How have past latitudinal diversity gradient analysis controlled for the effect of available area? That is, that 5 degrees cells or bands have much larger areas near the equator than towards the poles. Could this have driven the notion that there are more species in equatorial zones?
Yeah, this is a very good question. And we, Chaya in our paper in PNAS did look at area, but it wasn't significant. And one way of thinking about it is if both hemispheres have different areas of land and sea, then the latitudinal gradient should follow the area effect. But it doesn't because I think, um, you can have so many species increasing with area, but it reaches an asymptote. So after a certain time, you have more than enough area for all the species, and they're either very widespread or there's whole climate differences or something. So the effective area is not linear either, it reaches an asymptote. And so I don't think it affects it as much as we might think. And in fact, for terrestrial systems, sometimes smaller areas have more endemic species, so they have more unique species. So this is also an interesting difference with the ocean. We do see some high endemicity in the Southern Ocean because the bottom of South America, the bottom of South Africa, south of Australia and New Zealand have quite high endemicities because they're far apart in the ocean.
Okay, great. Next question from Adrienne Lam. Excellent talk. Mark, do you know of any studies that look at the Goldilocks zone temperatures for life as obtained or inferred from the fossil record?
Yes, there's a few studies we cite in our paper. They weren't exactly looking for 20 degrees, they just happened to find it. So we cited those studies. And I was really surprised about that. In fact, I think one of the referees of our paper in Trends in Ecologies was a paleoecologist, and he knew of this effect.
Hmm.
So he was very positive about our papers because it suddenly connected their fossil data to our living species data, which was kind of cool. But these are kind of only we're talking only a few studies so far, so we wouldn't want to be too certain yet, but definitely merits more testing.
Okay, great. Question from Bomsik Kim. I was wondering what kind of temperature index or standard a 20 degrees Celsius baseline refers to. Also, if it represents an annual average, I would like to know your thoughts on whether the duration spent around the temperature or the annual temperature range is also considered to be related to species richness.
Very good question. We've just used annual average temperature, and I am kind of surprised that it shows this relationship because there's things like degree days, which is the sum of temperatures over many days, is also important when you're growing animals and plants as well, actually, is more important than the actual temperature, is the kind of accumulated energy. So I think that does merit digging a little bit deeper into. Is it the range of temperatures? Is it the extremes?
Mhm.
Uh, so yeah, so I guess in biogeography we find this nice interesting pattern, and then we need to pull out the threads and see what's really the drivers at an individual species level. Yeah.
Great. Yeah, so a follow-up from Adrienne Lam is, in other words, do we know if 20 degrees Celsius stays stable in different time? But I guess that's what you just referred to when you talked about paleoecology and yeah, having some evidence for that.
Yeah.
Yeah,
Yeah, I think two or three paleoecology studies. Yeah.
okay, great. A question from Amy Geraki, and I'm sorry if I'm mispronouncing anyone's names. Um, could you speak further on your students' experience of using museum collections for historical data and correcting misidentifications?
Yeah. So razor clams, to me, they all kind of look very much the same. So when you look at the taxonomy, it's all based on the shape and the depth of the shell. So they're not the easiest group to separate. And so, but Hani specialized in them. She'd worked in them in Iran, where she did her master's. So she was kind of familiar with a few species and went on and looked at these. And I think in groups like that, and also there's a well-known problem that people in developing countries or Southern Europe first started using identification guides from the Northern Hemisphere. So they were giving people the same names as a Northern Hemisphere species because it looked almost the same. But they're not the same when you look at them again. So once these synonymies and taxonomies are sorted out, you actually see that the equator, at least in the ocean, I think is almost is a barrier to some extent for some species because it's just too warm. So they end up living mostly on each side where the temperature is just right. Yeah, I think museums are probably, and also the fossil specimens in museums, and of course, climate change, looking at distributions and what's changing over time. Another very interesting thing that somebody should look at is the Holocene climatic optimum. So around 6,000 years ago, the North Atlantic and the adjacent land was more than 4 degrees warmer than pre-climate change. So it's actually already been as warm as predicted under climate change by the end of this century. And I haven't seen anybody, only a couple of studies like at Svalbard showing that, oh yes, species are coming back and their shells were here from 6,000 years ago. So we're actually returning, all the species that have lived in this area have lived through this temperature somehow before. Um,
Yeah.
but some biogeography students should look at this, the pollen records, the shells.
The past can definitely be a great analog for what's going on right now.
Absolutely, absolutely. Underutilized.
Yeah. Richard Field asks, for the correlations of benthic organism species richness versus temperature, was the temperature adjusted to the depth? And more generally, can you test a 20-degree prediction by depth in the sea or in lakes?
Um, You probably could. We didn't do that actually. We used sea surface temperature. At the time we did that analysis, we found that the sea bottom temperatures available globally were a little bit variable. And I think it's just because of the way they had been measuring them, they didn't really capture the shallow depths, which is where most species are. So it was kind of convenient for us that most species are in shallow coastal zones, and that's where sea surface temperature is a good analog, or good metric. So, but now the environmental data sets are much better, the biodiversity data sets are much better.
Yeah.
So I think it would be possible to, and in our Arctic work, we found sea bottom temperature was a better correlation than sea surface in that case. So I, yeah, I think all this data keeps improving, so we should re-look at these hypotheses.
Okay, excellent. Another question from Iram Abbas. If we corrected for altitude at the exact collection point, not the grid average, would the terrestrial peak shift back toward 15, 18 degrees Celsius or stay at what you showed, basically, 10,
Oh,
15 Celsius?
great idea. We might try to do that, but that's an excellent idea. That's exactly what somebody should do. Yeah, we will actually look at that and see, because we have to. When we got the referees' reports back, actually, one of them had lots of good suggestions. So we will try and see what more analysis we could do, and that may be something to do. Yeah.
Okay, great. An anonymous attendee asks, thanks for the talk. How much does the spatial distribution of land and shallow shelf area control the gradient and influence the equatorial dip?
Almost not at all. I think either in the main paper or the supplementary material of Chowd Harry's paper in PNAS, she looked at area and continental shelf area as well. Yeah, and
Okay.
didn't think have that much effect.
Mark Lomolino, given the molecular energetic basis, do you expect analogous patterns for other major ecogradients, species elevation and species depth, for example?
Yes, maybe. It was such a big surprise to find this. And I don't think we were only driven to look for this 20-degree effect by the thermal energetics co-author who said he found this and he thought it was amazing across all domains of life. And did we see a signal at higher levels? So then we found all these examples. Um, and then, yeah, of course, individual species vary a bit, which can be confusing. But yes, I think we probably should.
Mm.
Yeah, yeah. Especially, I mean, like if humans, we can adapt. If 20 degrees is just perfect for us, why not for other mammals as well?
Okay. I see a comment in the Q&A. The beta and alpha peak is easy to explain. That's a comment from Olevetas, but no follow-up or a question. So take it for what, what you will. But there is a question from Kartik Trikadiri for terrestrial patterns. Do you think there is any reason to explore different biomes as separate systems? The marine terrestrial distinction is a strong one, so should we consider similar distinctions between, say, rainforests versus grasslands, especially in terms of the role that temperature precipitation play?
Yeah, we were kind of surprised that precipitation didn't show such. Well, the climatic zone, the ecological land units take into account precipitation and their pattern, and richness was correlated with them. So the more ecological land units have, the more species you have, as we would expect, in a latitudinal band.
Mhm. Okay. Um, we have we have time for a couple more questions, so let's just keep going. Another one from Richard Field. Also, given that temperature tends to be less variable the deeper you go in the sea, can you use that to disentangle the mean temperature and the variability of it?
Yeah, maybe. Yeah, yeah. I mean, temperature basically doesn't vary below 500 degrees except in a couple of places. Yeah.
Okay.
Mm.
Jordan Gold asks, thanks for the talk. Could you share some more thoughts on why turnover rate might peak at the equator while species richness declines in the bimodal valley? Valley in parentheses. Is this a consequence of a relatively rapid shift in species richness as you traverse the area between the bimodal peaks, or do other factors contribute?
Yeah, I don't know. So we just have to speculate into the discussion in the paper, but probably because we're looking at a latitudinal band, some parts of around that band are probably not changing and some are. So we're getting this variability around the equator, around the world. So probably you could find some places where there's no turnover and some places it's much higher, and we're looking at the average in that band. So I think this it's because the equator on average is very hot and you get extreme summers and or extreme events that you're causing species to be lost and maybe then some coming back again, increasing the turnover.
Okay. I'm really sorry. We are at time, so we really should cut this and let Mark go. Thank you so much, and I'm sorry if I didn't get to your question. Um, Mark, thank you so much for coming and giving this wonderful talk. Clearly, there's a lot of interest in LDG still, even though we thought we had it figured out. Seems like we might not have it figured out just yet, but that's great.
And
Lots
also,
of
when you get your papers almost rejected, which is what Hani Saidi did, we just said no. Let's look a little bit more at this, and then you find, you do a little bit extra work, and you find something that's a major finding.
Yeah, that's great. Persistent space, I suppose, is the message here.
Unappreciated skill.
Yeah. Thank you all very much for attending. I hope to see you all next month in August. Please join us again. We love to see you all here. So thank you again and till next time. Bye, everyone.